Hi everyone, welcome to Jack's Career Chats, a series where we highlight the diversity of jobs, skills, and opportunities in the world of biomedical research. I'm your host, Christina Valianatos. I'm the genomics educator at the Jackson Laboratory in Farmington, Connecticut. Today we're talking with Dr. Marina Yurieva, Bioinformatics Analyst in the Computational Sciences Department at the Jackson Laboratories campus in Farmington, Connecticut. Marina is trained as a scientist, and in her career so far, she's worked both in the wet lab, doing experiments with model organisms like mice and zebrafish, as well as the dry lab, using her bioinformatics and data science training to tackle big questions in biology and medicine. Marina, thank you so much for taking the time to join me today.
Thank you. Hi, Christina.
Hi. So you are a bioinformatics analyst. What does that mean? What is your job?
So it means many different things, but the main umbrella is I analyze all kinds of biological data. It can be data produced from the lab. It can be also already published data from the papers. I analyze it, creating different pipelines, writing codes. I integrate different data sets. So I analyze sequencing data, and basically there are different kinds of sequencing data. There is RNA-seq, DNA-seq, there is microbiome data, methylation data, all different kinds. And depending on the biological question we have, we can analyze it many different ways.
So it sounds like you get to do a lot of different things in your job. It's not just one monotonous thing. You have a lot of skills and a lot of projects.
Right, it's always different projects with different labs. I just transferred, to computational science department. Before I worked also at JAX with one lab, then it became 2 labs, and now it's 3 or maybe more labs. And I have projects with all of them, and they're very different. They don't really overlap, and there are many collaborations. Well, they ask all different kind of biological questions. They need help getting the data uploaded online for the publications. They need help with visualizations, with the data analysis, and all that.
So you have both laboratories that you work with at JAX, like traditional research labs, as well as companies, like external companies?
No, I didn't really work with companies. We collaborated with Yale and with University of Pennsylvania.
Oh, I see. Yeah. Other institutions, like other laboratories.
Other researchers, yeah.
I see. So it sounds like you have a lot of projects. How is it for you kind of juggling your daily tasks if you're working with so many different data sets from so many different labs? There must be very different projects, right? How is that for you?
Right, yeah. It's I've done something like that for a number of years, so now I think I have a grip on it, and I can compartmentalize it in my head, and I know which projects are the priorities and which projects I need to do today, who I need to talk to. It's a lot of emails, a lot of meetings. I attend the lab meetings for most of these projects. So it's a lot of balancing and setting up the priorities.
So you might not have then a typical work day, it sounds like, since you have so many different things going on. But what does your typical maybe week look like? How is your workload, and what are the types of things that you do?
Yeah, so different days of the week are different, and some days I have meetings most of the day, and I get to do the actual work only for maybe a few hours. Some days I don't have many meetings. I need to answer emails though. I collaborate with people, and mostly I set kind of a list of which things I need to do today and which deadlines we have, what is the priority, and try to work on that. So it's good in a way that for me it's not boring, and I can switch from one project to another. If something is very tedious, I can switch to a different project and then go back to the one I was working on later. So for me, it's good. Like my brain is very well set for that. But for some people who are not used to that, it might be challenging at first.
Sure, that makes sense. Maybe if you really only like focusing on one thing, this is not quite the path for you, not quite the job.
Right. Yeah.
So you mentioned that you have collaborators. Who are the types of people that you work with?
So I collaborate with, biologists, the people who I do the projects and the data analysis for. They are graduate students who are leading these projects, graduate students, postdocs, but mostly I communicate with PIs to see their whole vision of all of their projects.
So the PIs are the principal investigators, so the head of a lab, maybe a professor or someone that runs a research group.
Right. So PI would set a project and maybe different projects, and the PI will have the vision of how this project works together, and if there is any overlap, if what's going to happen. And the graduate student would have one of those projects and would lead one of them.
Yeah, so you mentioned that, maybe this isn't the best job for someone that likes to focus solely on one thing, but it sounds like a really good job for someone that likes kind of communicating with people, because you're communicating with a lot of different people, and you're almost like maybe this liaison. You're kind of taking, the information from the researchers, and then you're also taking their data and you're helping, communicate the information and convey what's there in their experiments.
Yeah, you definitely need to know many things. Like I have to know biology. I have to know programming, different programming languages. I need to be able to communicate fluently and freely to all kinds of different personalities, all different people. Yeah, it's interesting. I like it.
And the subjects that you work in, biology is a very broad area, right? So can you is there any focus, or do you have things in all fields, like immunology, neuroscience, cancer? Does it really touch on any field?
So my specialization right now is non-coding RNAs, regions of genome that used to be defined as basically junk because they didn't code any proteins, and for the longest time people thought that they don't have any function. But in recent years, there have been many publications and a lot of research that show that they actually have a role in cancer, in immunology, in brain development, in many areas of biology. So I work with non-coding RNAs. With another lab, I work on splicing. That's also RNA biology. That's that's how the gene gets modified and what version of the gene are expressed in different cells. So, for example, in cancer cell, you get the expression of one version of this gene, and in normal healthy cells, you will have the expression of a different version of the same gene. And so it gets very complicated and very interesting. That's kind of my specialization. before I used to do a lot of microbiome work, and I do some of it now, but not as much.
So you talked about what you did before. So can you talk a little bit about what your path was to this job? Did you always know you wanted to be a bioinformatics analyst, or how was it growing up, and what was your path like to this job that you have now?
Yeah, I would probably never thought that I would be bioinformatics analyst. I I decided, or I knew what I wanted very, very early in life. When I was 13, I already set my mind on being a scientist. And I decided that I'll be biologist because that's what I liked, and that's what fascinated me. I used to watch a lot of nature documentaries and read a lot of books about animals and nature. Uh, Also, my grandparents are scientists, so I spent all my summers in Siberia with them, and they would take me to their labs. Basically, I grew up in like nuclear physics lab and near organic chemistry lab. And it was fun. My grandfather, I don't know if he didn't realize that I was too young, but he made me type his scientific papers on the typewriter.
Oh, wow.
Yeah, and he would always talk to me like I was already a scientist and that I already knew nuclear physics. And I couldn't understand anything. He would talk to me about diffraction and everything else about nuclear physics. It was funny because it kind of boosted up my confidence that I can be that person. But honestly, I could not understand anything.
Do you think that influenced you, spending so much time in the lab and getting exposed to the science and having your grandfather talk to you about science that you didn't understand? Do you think that had a positive influence on your direction in science?
Yeah, I think so. I liked the way they lived and how excited they were about science, and they still are. I mean, my grandmother still works. Uh,
Wow.
yeah, so I liked the fact that there is so much unknown, and I really love the animals, and I love reading about it, and I couldn't honestly see myself doing anything else at first. Like I made the decision, basically grand decision when I was 13. Um, and it's a big decision to make when you're
Yeah.
young. Yeah, and over time it changed. So when I was 13, science was also very different. There was probably no bioinformatics, or it just started. And later on, when I went to my school, to the Moscow University, I went to the bioinformatics and bioengineering department. And that was very new. We were the second year that graduated from that department. Um, it
Wow.
was, yeah, it was very interesting. I think maybe even professors didn't know what to do with us. We got all kinds of different courses. Um, in Russia, you don't choose to take classes. You don't choose the classes that you want to take, and they're given to you. So we were given tons of biology, tons of chemistry, physics, um, math, like differential equations, linear algebra, and all kinds of different stuff. Uh, and I'm glad on one on one side I am glad. On the other side, I feel that it was really too much, and I don't use most of it right now. But that gave me the opportunity to learn how to learn. So to learn, to know where to look for stuff, to be open, and to know that there are other ways that you can look in science.
Yeah, I really like what you said about learning how to learn, because so much of science is discovery, and you're learning and you're researching, and you're in new territory. So even if you don't have a subject expertise, just knowing, the process of critical thinking and logical thinking and doing the research is very important, right? Even more so than the sub, or at least equally so than the subject expertise.
Right. And now what's happening is the integration of science, of different areas of science. And I think that that's very helpful. So you actually need to know math and biology and physics and chemistry all together to solve certain problems.
Yeah, and I think your career path has really illustrated that. So you started off as a biology interest in biology, and then it shifted, right, to the bioinformatics and
Not
computational.
really. So I started my bachelor's and master's. They were specialized in bioinformatics.
Okay.
But it was very different kind of bioinformatics that from what I'm doing right now. We didn't have any sequencing data. We were doing mostly comparative genomics. I didn't do tons of programming. I didn't really know programming languages, really. We were taught Java, and I never use it anymore. And so when I switched to my PhD, I I had that idea that I will do bioinformatics in a biology lab, but also I had to do the experiments. I had to work with mice, and it ended up that mice became most of my time. I was doing a lot of microscopy, a lot of experiments with mice, with mouse embryos, and I did a lot of data analysis, but probably 60-70% of my time was the work in the lab.
And how was that transition? Because working on a computer, kind of the dry lab side of things, and working in the lab, experiments, with model organisms, those are very different things. How was that transition? How was that balance?
I was terrified.
Why?
Because I actually didn't really work in the lab before my PhD that much. I had some projects, but working with mice is on its own very specific thing to do. Mice take very long time, and I was working with mouse embryos, so you have to wait for the mouse to get pregnant and have the embryos, and all those experiments would take weeks. And for me, I want everything fast, and I wasn't ready for 2 weeks of experiments that sometimes wouldn't work. So it was challenging, but I'm really grateful for that experience because I got to work with microscopy. I used to work on laser dissection microscope. That was really cool. I would dissect out a small, very tiny part of the brain of the mouse embryo, and put it on the slide, go to the laser dissection microscope and dissect it out with the laser.
An even smaller part.
Yeah, it's smaller. It was very small. It was very, very tiny. Yeah, it was cool. And then I got to work with zebrafish embryos and with chicken embryos. It was all very helpful to see how biology works in very different models.
And what motivated you to pursue a PhD? So you did your undergraduate training, and why did you want to go back to school and to get your graduate degree?
I didn't know what else to do. I mean, you are already set your mind on being a scientist. And I think also there weren't many different career paths at that time. There were different options, but for me, it was important for me to learn more. I knew that I didn't know everything, and certainly I don't know everything now. Um, so it was important to learn, and it was something that I don't know. I didn't really think about not doing that.
So it's like you just knew kind of as a scientist, this is the path that you have to take in order to be a scientist. This is what you do.
Right, right, yeah.
So you mentioned that kind of your first bioinformatics training was in things that you don't, some of the things was in things that you don't really use now, like, you know, Java programming language. So the things that you do now for your coding and programming and analysis, where did you learn that? Did you learn it along the way? Did you have formal training? Like what was your training like?
I never really had a formal training. I had some courses I would take um, online, or I went to one course in Seattle once. But mostly it was self-learning. It was, you get the project and you need to do something. Usually you need to do something fast, so you need to learn. It's the needs that drives it. I knew a little bit at my PhD. I was analyzing microarray data, and then when I went for my postdoc, that was an actual bioinformatics position. I was working in a computational facility, in bioinformatics facility, and I was like I'm doing what I'm doing now. I'm analyzing different data sets for different labs. That's what I was doing then too, except I didn't know what I was doing. I had to learn all the Unix environment. I had to learn R much better than I used to know it, Python, and I had to learn how to do everything really fast.
Well, I think you bring up such an important point because, you know, you got a job, but you still didn't know all the skills, but you were able to learn them in the job. And I think so many, you know, young scientists or students think, oh, I have to know all the things in order to, you know, get this job and be really good at it. But it sounds like, you know, oftentimes there's some wiggle room and you can learn along the way. You're expected to learn along the way. Is that the case with you? That's what it sounds like a little bit.
I think I had very good bosses and very good supervisors that um, maybe saw some potential in me, and they saw that I could do it before I saw it myself. And sometimes it would be like for my postdoc, I came and I was like, can I even do this? I don't know. I'm gonna get fired. I I'm not that person. And then I learned, and it was scary, but it wasn't that hard. You know, the hardest part is that the fear.
The fear you put in yourself.
Right.
Yeah, that's a really good message, you know. I mean, well, there are two messages there. One is like the good mentorship and the good supervisors, right? And how important they can be in your journey and supporting you. And then
Yeah,
another,
yeah. People who you work with are always important, and especially as supervisors. I think maybe sometimes younger people, they don't have that motivation yet. They don't know what they can do. And my PhD supervisor was just brilliant. He was that big famous guy, and he he was also the scientific director of the institute, so he was busy all the time. But every time I would go to meet with him in his office, I would close the door, and then he starts talking about science, and he's so almost obsessed. And he tells you all his ideas, and it can last hours, literally hours. It might not be even about your project, but he would see something in your project, and then he starts talking, and he's all excited. And it was so cool. It was, I wish I had that motivation, but he kind of inspired me to do that.
Yeah, I think sometimes that can be very intimidating when someone just like rolls with the ideas so much faster than you. But I think, taking that as inspiration instead, seeing the bright side of that and being motivated by that, that's a great thing.
Yeah, of course, you cannot implement all the ideas your bosses tell you to do, because sometimes there are millions, and you can pick something that is really worth looking at, or something that you think gonna work, and go with that.
Just to shift gears a little bit, this past spring, when you were in your position, you pretty much put everything on hold. And during the coronavirus pandemic, you volunteered in the JAX CLIA lab, the COVID testing lab. Can you talk a little bit about what motivated you to do that and what your experience was with the COVID group?
Yeah, it was great. I really enjoyed doing that. And in my life, besides my work, I do a lot of volunteering. So it was great to be able to volunteer at JAX. And it was good that somebody needed my help, and I had the skills to do that. It was also very interesting to see that big difference of CLIA lab, the clinical lab, versus the research lab. Clinical lab is very organized, it's very structured, and everything has to be approved. All the reagents, all the protocols. The protocol itself is, it's just regular qPCR. But every step of the protocol had to be approved. There are different people who do each step of the protocol, and there was great attention has been paid to the labeling of the samples. So the issue was that we get the samples from different individuals, and we cannot mix them up. So there were like 3 people who would double and triple check the labels.
Wow. So it sounds like a lot of extra organization. Not to say that normal research, your typical research lab isn't organized, but the clinical lab was kind of extra organized because of dealing with the patient samples.
Right. And it makes sense, because in clinical lab, we already have the protocol that we already know works well. And it has to be approved by FDA, by the government. In research lab, we don't really know much. And if we're studying something new and we haven't tried it before, then we need to discover, we need to change protocols, we need to change temperatures and the time and everything else. So of course, the research lab cannot be structured like that.
Yeah, much more rigid, less flexible.
Yeah.
So were you also doing bioinformatics analysis for the clinical lab, or what were you doing when you volunteered?
No, I was doing a part of the protocol. They already had, I think, the research IT and the computational sciences departments. They took the analysis of the samples and the results. And I think they developed many pipelines and databases. They developed a big database that the people in the lab could input their samples, and they can communicate with the hospitals and the clinicians about the samples. And that was a lot of work. Yeah.
And your role during this time was in the lab?
I was just unpacking the samples and putting them together and double checking the labels. So it wasn't what I would normally do.
Yeah, I think that's an important point to bring up, because it is very different than what you normally did, but you know, it was put towards this great effort of increasing the testing in our state and helping with this pandemic.
Right, yeah.
Can you tell me a little bit about your mentorship and, you know, as a woman in science, did you have female mentors? How was that for you in terms of, you know, just representation of having someone that looked like you and thought like you in the lab as a mentor?
No.
Yeah.
I knew other female scientists. They were not, all my bosses were always men. Right now I have one woman I work with, but it didn't happen until now. So it was, I think, especially for computational biology, it's a big issue that you go into the room for a lab meeting, for a meeting of the department, and most of the people there are men. And for me, it was terrifying at first. It was like, what am I doing here? Am I even allowed to be here? But I think some of it is that internal fear that I had, that I really need to have confidence to equate myself to the men, that I'm not any less than them. And they, most definitely, none of the people I worked with made me feel that I'm less than them. Mm-hmm. So that was really good and encouraging. And I had to learn that I can do as much or maybe more as them. And that's for me. For me, I don't know how it is for others, but for me, it's more like my personal issue of confidence. And there are other issues in being a woman in science that are not really about that you cannot do much about that. You know, I'm not a hiring manager. I cannot hire more women. I just not allowed to. If I could, I would. But it's right now, it is the reality that we don't have many women in science, especially computational biology. We don't. Being a woman in science and being gay and being a foreigner. And I think right now the focus is targeted mostly to being a woman in science, but not so much towards being gay or a foreigner, because it's not very science specific.
Yeah, I see. So how has your gay identity impacted your scientist identity? You know, how have those worlds combined? I mean, for you, they are one world, of course, but are there any unique challenges or advantages to that perspective?
I think there is nothing special about, like, I'm not a gay scientist. I don't really write gay pipelines, but it's more about visibility. That I don't know many other gay scientists. And also, as a foreigner, it was my decision to not really be public about it for many years, and not to talk about it, because coming from a country that has a lot of homophobia, and that you know this kind of video would be banned in Russia right now.
Mhm.
So I I didn't really know or understand that you can be open like other people here, that you can just freely talk about your weekend. Um, and right now, I think I choose to be more visible, and it does have its own consequences. So as being gay, as being a foreigner, I have to deal with some uncomfortable or inappropriate questions a lot. And, you know, I can either choose to, I can choose not to be a foreigner. So this is what people, or not be a woman. This is what people will see anyways. And I will have uncomfortable questions about that regardless if I show that or not. But I can choose to never talk about my personal life. And it's not really, I think, the choice I want to make now. I
Yeah.
think, yeah, this, like right now, the time is changing, and I, I think what I can do is to show people that they can learn how to talk to the minorities, that it won't sound offensive. I mean, you can Google that. You can Google what kind of questions you don't ask foreigners, or making jokes, or like how many people told me, "Oh, you don't drink, not even vodka," and yeah, it's supposed to be a joke, but it's not really.
Mhm.
Or I have to talk about bears and the president, and I I'm not I'm not responsible for that.
Yeah, absolutely. And as you said before, along the lines of what you were saying before, our identities are complex. We are not just one thing or two things, and it's not just the stereotype version of that, right? And so, we're all very well-rounded, complex, different people. And I think, your visibility and being so open and honest, while it may make you vulnerable and expose you to these types of, questions, inappropriate comments, things like that, it helps for especially like the next generation, younger scientists, anyone, student, anyone looking to fit in or looking for their path. It's so important because we're all so complex, and there are so many barriers in science and in life. And so seeing someone represent you and represent your beliefs and your path and your upbringing, I think is so
Right,
important to help you see that you can do it and you can be there.
right. And I don't really want it to be labeled as open and honest, because it just who I am. I don't. It's not a confession.
Right, right.
I want it to be a regular, normal life, and I hope that people who are watching, they are the new generation, and that for them it will be different, and that for them it will be just normal, and they don't have to have all those hard conversations about being a woman in science, or being a woman in general, conversations about Me Too movement, about being a foreigner, about being trans or gay or anything else. And that people learn from that, and people learn to be more kind to each other, you know?
Yeah, absolutely. And just to have, representation and to just have more visibility. And yeah, I think, when I said open and honest, it's more just because you don't have to, right? Because some people might think that, oh, I have to hide who I am so that I fit in, or that I fit a certain mold or a certain stereotype of what it means to be a scientist or to do science. And I think what you're saying and what you're showing is that, you should be yourself, and everyone is welcome. Science is a diverse field. The world is diverse. We should have everyone here, and everyone should be represented.
Yeah, and right now it's definitely a choice. I don't um, tell anybody you should tell everybody that you're gay or that you're trans or anything else. It's a personal choice. Um, I think as a foreigner, when you come from a country like that, there is a lot of fear that's instilled in yourself that I I cannot talk about it, and that, like, it's still all the culture and all the stigma, it comes with you. It's a part of your history, and it's hard to just get rid of it by moving across the border.
Yeah, no, that makes sense. I mean, you're carrying everything with you to this new place, and you're learning to adapt and to
Right.
carry with you what you have from your own upbringing.
Yeah, that's true. And there are certain things about being a foreigner in science that are out of our control. It's more the government, it's the immigration issues, it's
Sure.
the visas and
Funding opportunities.
funding opportunities. Yeah, it's that you have to be basically stuck here for several years sometimes, because you cannot go back to your home country, because getting a visa is such a process that um, you will have to wait for 2 months or more, or you don't really know how long, but they do that background check thinking that you're a Russian spy. Um, and yeah, I mean, it's true. It's funny, but I have to. Um, I heard many jokes about myself that you're probably a spy, and sometimes it was serious. And I know that the government thinks so, because they check me every time. And what can I say? I can't say I'm not a spy, because the more I say it, the more people will think that.
Yeah, I'm so sorry that you've had to go through that.
It's thank you.
But I'm sure there are many, kind of good things about being here. You're choosing to stay, so hopefully that the opportunities, here, as well as the work that you do and the impact that you're making in the research outweigh some of these more taxing and difficult scenarios.
Yeah, of course. It's the opportunity to work with great people, and the funding. It's hard to get the funding, but it is there. It exists. In some other countries it doesn't exist. It's very smart people. It's good atmosphere, and it's I think the the main goal of everybody to make the science better.
Yeah, absolutely. And that's why we need so many different types of people with different experiences and different backgrounds to make the science the best it can be, right?
Right.
Do you have any kind of parting words to share with some aspiring scientists or young students that are looking for careers in in bioscience, in bioinformatics, computational biology?
Well, it's important to have confidence in yourself, and I think it's important to not let anybody make you believe that you cannot do something and that you're not good enough for something, because nobody really knows. Nobody knows your life. Nobody knows what you are actually capable of. And for me, it personally stopped me from being more motivated, work harder. It was, that here it's not pleasant experience. And I think I was lucky that I kind of chose my career so early on, and that was my internal motivation, that I knew that I really wanted to do something, and that would wake me up every day. And I hope that everybody who sees that, they do have that motivation. It doesn't matter what it is. It doesn't matter if you really don't know what you want to do, but you can be open, you can be curious, you can learn new things and eventually discover a thing that you really like. But it's important to build that resilience and to not let yourself being broken by failure, and more learn from it. If something didn't happen, if I couldn't do that, what can I learn from it? What was the message? When I look at it that way versus I failed, I can never do that again, it's over, then I can learn more.
Yeah, absolutely. That's wonderful advice, and it's very inspiring to just kind of stay driven and keep motivated and be resilient.
Yeah.
Well, thank you so much, Marina, for joining me in this chat. It was really nice to talk to you. I learned a lot about your career path and the job that you have now. It was really great.
Thank you for having me.
And to our viewers, thank you so much for tuning into this episode of the Career Chats. And don't forget, we have plenty more interviews to watch on our Career Chats series. Until next time, goodbye.